Nature's Blueprint, How Ancient Animals and CRISPR Could Cure Aging. All right, welcome to today's Explainer. We're about to go on a seriously fascinating journey. We're going to start in the freezing, pitch-black depths of the Arctic Ocean, and we're going straight into the microscopic, bustling world of human DNA. We're looking at absolutely nothing less than nature's ultimate blueprint for survival. And more importantly, we'll see exactly how modern science is trying to borrow that blueprint to literally redefine human aging.
400. Okay, I want you to just imagine an animal swimming in our oceans right now that was actually born before the invention of the telescope, before the English Civil War, even before the Taj Mahal was built. That is the estimated lifespan of the Greenland shark, up to 400 years. It absolutely shatters our whole understanding of biological limits, right? And it really ignites this massive question. What exactly do they know about aging that we don't?
Section 1. Secrets of the Deep. Let's dive right in.
So to really understand how we might cure human aging, we first have to look at the longest living mammals and vertebrates on the planet. Think about us. An average human lives about 80 years, right? And we carry a genome of roughly 3.2 billion base pairs. Now contrast that with the Bowhead whale, which casually lives over 200 years, and our friend the Greenland shark, hitting that staggering 400-year mark. But here's what's incredible. The shark's genome size is 6.5 billion base pairs. That is more than double ours. Now usually, scientists look at massive repetitive DNA sequences and think of them as selfish or even harmful, just because they don't seem to carry useful information and they can cause genetic instability. But in the case of the Greenland shark, these repetitive elements are actually a massive clue. It turns out, this giant genetic code is packed with duplicated genes that are entirely dedicated to keeping the animal alive and intact for centuries. It's like a built-in redundancy system for survival.
Section 2. Masters of DNA Repair.
You know how throughout our lives, our DNA constantly accumulates damage. It's kind of like typos in a really long manuscript. If those typos aren't fixed, they turn into permanent mutations, and those lead straight to aging and cancer. So let's look at how the Bowhead whale handles this. They produce a specific protein called CIRBP. Because these whales spend their entire lives in freezing Arctic waters, this cold-triggered protein is produced at levels 100 times higher than in humans. Literally 100 times. And what does CIRBP actually do? Well, it faithfully repairs severe DNA damage, specifically those really tricky double-strand breaks in the DNA helix. Here's the wild part, though. When researchers took human cells in a lab and boosted their levels of CIRBP, the human cells actually doubled their ability to repair DNA. No way, right? So now scientists are actively looking for pharmacological ways to mimic this effect in humans because, let's be honest, as amazing as the science is, I'm guessing you don't really want to take a daily Arctic ice bath just to boost your proteins.
Now, moving back over to the Greenland shark, they have their own unique tool. It's an alteration in a protein called p53. In the biology world, p53 is famously known as the guardian of the genome. It's basically the most important tumor-suppressing protein we know of. It coordinates exactly how a cell responds to DNA damage. While the Greenland shark has unique structural alteration in its p53 protein that makes it just incredibly efficient. This really cements our understanding that the animal kingdom's longevity toolkit isn't magic. It's all about relentless, highly optimized genetic maintenance.
Section 3. Enter CRISPR gene editing.
So nature has clearly already solved the aging problem. The real question is, how on earth do we copy it? And this is where we dramatically pivot from marine biology straight into human genetic engineering. As Professor Vera Gorbunova, who's a leading biologist in this field, points out, people used to think we can't improve DNA repair, that it's already optimal. But the whale does it better than we do. Guys, this is a massive paradigm shift. For decades, we really thought human biology had just reached its peak evolutionary limit. But we're actually no longer bound by those biological limits if we can just learn from these whales and sharks. We just need the right tool.
And the tool that allows us to bypass those limits? That's CRISPR-Cas9. You can basically think of CRISPR as a pair of programmable molecular scissors. It works in 3 pretty simple steps. First, we engineer a guide RNA, or sgRNA, which acts exactly like a GPS, to find the precise genetic typo in the DNA sequence. Second, the Cas9 enzyme, that's the scissors, snips the DNA at that exact location. Third, the cell's natural repair mechanisms kick in. From there, we can either let it repair via a process called NHEJ, which effectively knocks out or just deletes a bad gene, or we can provide a healthy DNA template to trigger what's called HDR, which completely corrects the mutation. We can literally paste in the healthy code. It's absolutely wild.
Section 4.
treating age-related diseases.
Now, while living to 400 years old sounds pretty cool, what really matters right now is treating the devastating age-related diseases that are affecting our loved ones today. Now, what's really interesting about this slide is how we can map this abstract genomic science to tangible real-world diseases. For Alzheimer's, we can target specific mutation genes like APP and PSEN1. For Parkinson's, we target genes like LRRK2. And for cancer, massive drivers like TP53 and KRS. We are no longer just putting a Band-Aid on the symptoms of these diseases. CRISPR allows us to go to the absolute root cause, the actual source code, and just rewrite it.
Let's look a little closer at neurodegeneration. In Alzheimer's, the disease is largely driven by the buildup of these toxic amyloid beta plaques in the brain. By using CRISPR to actually correct mutations in specific genes, scientists have successfully disrupted the production of these toxic proteins in lab models. That's huge. Then in Parkinson's, the issue is really often tied to the accumulation of damaged mitochondria, the powerhouses of the cell. Using CRISPR to edit specific regulatory genes can actually force the cells to clear out those dysfunctional mitochondria. We are literally talking about potentially replacing years of cognitive decline with healthy lucid years.
And then, of course, there's cancer, which is perhaps the ultimate disease of aging and unchecked cellular mutation. CRISPR is being used to quite literally disarm cancer cells. By knocking out specific oncogenes like PAR3L in colorectal cancer or DMMT1 in ovarian cancer, researchers are seeing something absolutely amazing. Not only do the tumors just stop proliferating, but knocking out these genes strips the cancer of its defenses entirely. It makes the tumors highly sensitive to traditional chemotherapy. The cancer cells essentially undergo programmed cell death. It's a real game changer.
Section 5. Challenges and the future.
Now, as incredibly exciting as this all is, we really have to ground ourselves and address the scientific roadblocks standing between the lab and the pharmacy. Keep in mind, gene therapy is permanent, which means we have to get it perfectly right. First up is delivery. How do you get the CRISPR scissors exactly where they need to go, especially past that heavily guarded blood-brain barrier for a disease like Alzheimer's? Right now, we often use viral vectors, like AAVs, to ferry the CRISPR tech into our cells. But our bodies are smart, and sometimes our immune system just attacks those viral delivery trucks. Second is dosage and off-target effects. If the dosage of Cas9 is too high, the molecular scissors might get overactive and actually cut a healthy piece of DNA by mistake. And finally, there's translation. Just because we successfully knock out an Alzheimer's gene in a cell doesn't automatically mean the patient's memory suddenly comes back. We have to definitively prove that editing the gene reverses the physical symptoms.
But listen, science is moving at a breakneck pace, and the solutions to these hurdles are literally being developed as we speak. We are standing on the precipice of a totally new era of human biology, one where we can look to the freezing, deep ocean to rewrite our own genetic destiny. Which really leaves us with a profound question to close out today's explainer. If we successfully clear these hurdles, and we can actually borrow the blueprint of a 400-year-old shark to edit away age-related disease, how many years are you prepared to live? Seriously, think about that. Thank you so much for joining me today, and as always, keep questioning the world around you.